Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. If you want to stay on top of the latest breakthroughs that are truly shaping our understanding of the world, make sure to like this video and subscribe to our channel. Your support really helps us continue to bring you these deep dives into cutting-edge research, and we truly appreciate it. Today, we're embarking on a really fascinating deep dive into a new study. It's completely reshaping how we think about type 2 diabetes, or T2D. Now, for decades, we've known that protein misfolding is, well, a major culprit in T2D, especially within those pancreatic beta cells, the incredibly vital cells responsible for making insulin, the hormone that regulates our blood sugar. But for a long time, the scientific spotlight was pretty much fixed on one specific part of the cell as the main site of this protein trouble. This new research, though, just published in Nature Metabolism, it's not just adding a piece to the puzzle. It feels like it's turning the whole puzzle upside down, pointing somewhere else entirely.
That's precisely right. Yeah. For years, the main hypothesis, and it was backed by solid research, it really centered on protein misfolding, happening largely within the endoplasmic reticulum, or the ER. You sort of think of the ER as the cell's busy factory floor, right? Where proteins get assembled, folded up, prepared for shipping out. But this new study, it uses these incredibly advanced proteomics techniques, allows researchers to get a really detailed look inside human pancreatic islets, specifically from T2D patients. And what they found is this robust, undeniable accumulation of misfolded proteins, but specifically within the mitochondria of these cells.
The mitochondria, not the ER.
Exactly. And this isn't just a minor shift, It's a crucial pivot in how we understand the very progression, maybe even the origins of T2D. It really changes the game.
Okay, let's unpack this then. So we knew protein misfolding was involved in T2D. And previously, the thinking was, well, it's proteins like proinsulin, maybe islet amyloid polypeptide, IAPP, misfolding in the ER, and that misfolding leads to stress, beta cell dysfunction, eventually beta cells dying off. So what's so different now? Why does the location matter so much?
Well, what's really fascinating is how they pinpointed this new location. The researchers, they didn't just look broadly. They did these incredibly precise quantitative proteomics studies right on the actual protein, aggregates those clumps of misfolded proteins you find in human islets. And the molecular signature they found inside those clumps, it just screamed mitochondrial protein misfolding much more strongly, much more consistently than anything pointing to the ER.
Wow.
And this isn't just moving the problem from, say, the factory floor to the break room. If the ER is the cell's shipping and assembly line, the mitochondria are the powerhouses. They generate nearly all the energy the cell needs. So a problem there isn't just a glitch in making something. It's a direct hit to the cell's ability to power itself, to actually do its job.
Okay, so the location is definitely a key distinction. And if the mitochondria are the powerhouses, what actually happens when their own internal proteins start misfolding? What's the fallout for those crucial beta cells?
Oh, it has huge implications, really significant cascading effects. Beta cells are, well, they're hyper-dependent on healthy mitochondria. These organelles are absolutely vital for everything from sensing fuel like glucose to precisely controlling insulin release, even just maintaining the number of beta cells you have.
So they do a lot more than just energy production.
Absolutely. So when proteins inside the mitochondria misfold, it's not just a small error. It leads to pretty profound structural defects, like the mitochondria literally get misshapen. And it causes respiratory defects. They just can't generate energy efficiently anymore.
Okay.
And that dysfunction, it fuels oxidative stress, the damaging imbalance of free radicals, and ultimately it triggers beta cell apoptosis.
Programmed cell death.
Programmed cell death, exactly. And this failure, this loss of beta cells, is precisely what then drives the hyperglycaemia, the high blood sugar. That's the hallmark of type 2 diabetes.
So it's a direct line, really. Misfolding in the mitochondria leads to cell death, leads to T2D symptoms.
It appears to be a very direct and damaging pathway, yes.
Okay, this is where it gets, I think, really interesting. The study points to a specific player in all this mitochondrial drama, a protein called LONP1. What exactly is LONP1, and what did they find out about it in T2D?
Right, LONP1. It's an absolutely crucial player here. It's a mitochondrial matrix protease. You can think of it like a specialized quality control enzyme, maybe a cellular recycling agent, but operating deep inside the mitochondria.
Okay.
Its main job is to recognize, grab onto, and basically turn over or degrade misfolded or damaged proteins. It keeps the mitochondrial environment clean and functional, essential for protein quality control.
Sort of the mitochondrial cleanup crew.
Exactly. That's a great analogy. And what they found, which was quite unexpected, was that the expression of LONP1 is significantly reduced, lower levels.
reduced in T2D.
Yes, reduced in the beta cells from human T2D donors.
Mm-hmm.
And this reduction is a major issue because it directly messes up the cell's ability to maintain optimal protein homeostasis, what scientists call proteostasis, within the mitochondria.
So the cleanup crew is understaffed.
Precisely. And that leads to a dangerous buildup of, well, junk, misfolded proteins.
Okay, so if less LONP1 means more misfolded proteins piling up in the mitochondria, leading to all those bad things you mentioned, like poor energy production, cell death, high blood sugar, the obvious next question is, well, what if you increase LONP1?
Did they try that?
They did, yes, and this is where the findings get incredibly promising, potentially for future treatments. They didn't just observe the problem, they intervened. When they induced what's called a gain of function for LONP1, basically, boosting its levels and activity.
They made more of it. Essentially, yes. And they saw remarkable improvements. This increase in LONP1 significantly ameliorated or, improved the mitochondrial protein misfolding problem. And really importantly, it restored human beta cell survival, even when those cells were put under really harsh conditions.
Conditions like in T2D.
Exactly. Conditions that mimic the metabolic stress of T2D. They call it glucolipotoxicity. It's when cells are just overwhelmed by high sugar and high fat levels, like you often see in uncontrolled diabetes.
And boosting LMP1 protected the cells even then.
That's what they found. A very encouraging result.
Now, usually when we hear protease, like LMP1, we think of something that, cuts up proteins, breaks them down. But the study suggests something maybe more subtle, even surprising, about how LOMP1 is actually working here to protect the beta cells. Is that right?
Precisely. Yeah, this is a really key insight. One of the most exciting parts, I think. LOMP1 is actually a multifunctional protein. And while it does have that protease activity, it can degrade proteins. The study found that its ability to promote beta cell survival and prevent that mitochondrial protein misfolding. It seems to be driven primarily by its chaperone-like activity, not its cutting action.
Chaperone-like, so like helping other proteins fold correctly.
Exactly. Think of a chaperone protein as more of a guide or a helper. It doesn't just destroy the bad ones. It helps other proteins fold correctly right from the start, or it helps refold ones that have gone a bit wrong. And this study showed really convincingly that LOMP1 partners up with another important mitochondrial protein. It's called mitochondrial HSP70 or MTHSP70 to do this folding job.
Okay.
And crucially, they showed this effect was entirely independent of its protease function.
How did they show that?
Well, they were clever. They made a special mutant version of LONP1, one that completely lacked its ability to cut proteins. Its protease activity was gone. And even that version, the one that couldn't cut, could still rescue the beta cells from apoptosis.
Wow. So it's not the cutting, it's the helping to fold part that's protecting the cells.
That seems to be the primary protective mechanism in this context, yes.
So it's less like LONP1 is the garbage disposal, chopping things up, and more like it's the quality control inspector, or maybe the tailor, fixing things or helping them get made right in the first place.
That's a great way to put it. It's a significant shift in understanding its main job here, especially concerning T2D.
That really changes how you might think about fixing the problem, doesn't it?
Absolutely. It suggests we might not need to focus only on getting rid of bad proteins, but maybe on boosting the cell's own ability to assemble and maintain proteins correctly.
The study also seems to build a strong case for the order of events, the chronology. Is this mitochondrial protein misfolding an early warning sign, like the first domino to fall, or is it something that happens later on?
That's a critical question, yeah. And the findings really point towards mitochondrial protein misfolding being the initial insult in this pathway. They gathered pretty compelling evidence. They saw mitochondrial protein misfolding happening in mice that were deficient in LONP1 before any other signs of metabolic problems showed up.
Before blood sugar issues.
Before changes in glycemic control, yes. Before changes in how well the beta cells released insulin in response to glucose. Even before noticeable changes in the total number of beta cells or their survival rate.
So it's happening really early on.
Extremely early. And critically, it also seems to happen before significant oxidative stress builds up and before you see major defects in those electron transport chain complexes.
You mean complex 1, complex 2, 3, 4, and 5, the energy machinery.
Exactly. Those core components within the mitochondria that generate most of the cell's energy. The protein misfolding seems to precede the breakdown of that machinery and widespread oxidative damage.
Okay, so that implies that while oxidative stress definitely is a problem in T2D, it might not be the absolute first domino in this particular chain reaction. It might be more of a consequence of the mitochondrial protein folding issues.
That's what this evidence suggests, yes. The protein misfolding appears upstream.
Which then leads to the next big question. Why is LOMP1 reduced in the first place in people with T2D? Is there something controlling LOMP1 that's going wrong?
That's a really important question, and they did look into it. The researchers investigated potential upstream regulators, things that might control how much LOMP1 gets made. They looked at transcription factors, those proteins that act like switches to turn genes on or off.
Right.
And they found a significant reduction in one specific transcription factor. It's called ATF5. And this reduction was seen in the beta cells from human T2D donors.
ATF5, and that controls LOMP1.
Yes, ATF5 directly regulates the LOMP1 gene. But it gets more interesting. ATF5 also plays a key role in something called the mitochondrial unfolded protein response, the MUPR.
The cell's own defense against mitochondrial stress.
Exactly. So the system the cell uses to cope when things go wrong with proteins in the mitochondria. So if you have less ATF5, you likely get less LOMP1.
And you also might have a weaker defense response when things do go wrong.
Precisely. So reduced ATF5 could be a major piece of the puzzle, contributing both to the lower LOMP1 levels and maybe to the failure to properly activate these crucial stress responses in T2D. It looks like a potential root cause, or at least a key contributor.
So putting this all together, what does this mean for, someone trying to understand T2D better, or maybe even for developing new treatments down the line? It feels like we're moving beyond just thinking about blood sugar or even just the ER.
Well, connecting it to the bigger picture, I think these findings give us really crucial insights into the specific kind of proteotoxicity, the protein-related damage that drives beta cell loss in T2D. It really highlights that T2D is, even more complex than we thought. It involves multiple systems for maintaining protein health, not just one.
Multiple proteostatic pathways, right?
Exactly. And the really promising effects they saw when they activated LOMP1, even potentially pharmacologically, that suggests entirely new therapeutic targets.
So intervening much earlier in the process.
Potentially, yes.
Yeah.
Targeting the root cause of the mitochondrial dysfunction, rather than just managing the downstream consequences like high blood sugar.
It really seems to connect with what we're learning about other conditions, too, like aging-related diseases, where problems with mitochondrial health and protein folding are known factors. It gives us a new angle, a new molecular lens, maybe, to view T2D through, and potentially new ways to treat it, maybe even prevent it.
Indeed. The whole idea that we could potentially activate these mitochondrial proteases and chaperones like LOMP1 to actually improve how efficiently beta cells work, their metabolism, their viability, and use that to treat or maybe even prevent T2D. That's moved from pure speculation to something that's, an intriguing possibility backed by solid data. It's definitely ripe for future exploration.
It really changes the perspective on maintaining beta cell health.
It certainly does.
And for you, our listener, I think this deep dive really highlights how science is always moving, always refining our understanding of complex diseases like T2D. It shows how research can shift our focus to, unexpected places, like the mitochondria in this case, revealing new pathways and new possibilities. It just underscores this incredibly intricate dance of proteins inside our cells and how keeping that balance is absolutely critical for our health. That's all the time we have for this deep dive into mitochondrial protein misfolding in type 2 diabetes. Thank you so much for joining us on the SAM Science Experience. If you enjoyed this exploration of cutting-edge science, please remember to hit that like button and subscribe to the SAM Science Experience for more impactful research breakdowns and fascinating scientific discoveries. Your support truly helps us bring you these valuable insights, and we look forward to having you with us on our next deep dive.